Hello volunteers! Welcome to this brief tutorial to help you participate in the Monkey Health Explorer project. The main objective in our project is to count and identify white blood cells in rhesus monkeys. This will help us understand which monkeys may be sick and which are healthy. There are 5 types of white blood cells. How can you tell them apart? How can you tell which one is which? In this tutorial, we'll describe some distinguishing characteristics that you can look for in a white blood cell to help you identify it. Let's get started. Each of these 5 white blood cells illustrates a unique characteristic. As we work through each of these slides, please note that preparation and staining techniques change from year to year, so the overall coloring and darkness of the images might vary from slide to slide. First, you'll notice that the majority of cells found in the images are actually red blood cells, like those pointed out here. You'll also see platelets. They're smaller, rounded shapes, and they're typically the size of the hole in the donut-shaped red blood cells. The cells we're focusing on in this project are the white blood cells indicated here: neutrophils, eosinophils, monocytes, lymphocytes, and basophils. Let's start with the most common white blood cell, the neutrophil, along with the less common eosinophil. We're showing these together because they have similarities that can cause them to be mistaken for one another. Here are some additional examples of each type to show the variation from cell to cell and slide to slide. Both of these types of white blood cells are typically 2 to 3 times as large as red blood cells. They both have multiple dark shapes inside the cell, which are lobes of the cell nucleus. Neutrophils tend to have more than 3 of these dark lobes linked together like beads on a string. In contrast, eosinophils typically have only 2 or 3 linked lobes. They sometimes look like a pair of lungs. Another distinguishing characteristic is the relative darkness of the cell's cytoplasm. The cytoplasm of neutrophils is typically a lighter shade, while that of eosinophils is typically darker. This is caused by small granules in the cytoplasm that make it look more shaded. Some slide images are darker than others, which can make it more difficult to assess the darkness of the cytoplasm. One way to help identify this is to compare the darkness of the cytoplasm to an adjacent donut-shaped red blood cell. The shading of the neutrophil cytoplasm looks more similar to the inside of the donut, while the eosinophil cytoplasm tends to be as dark as the outer solid part of the donut. One more factor to consider is how common the cells are. In humans, neutrophils make up 40 to 60% of white blood cells, while eosinophils make up only 1 to 4%. These values come from studies using human blood because we don't yet have a standard range for rhesus macaques, but we expect them to be somewhat similar. Now let's compare monocytes to lymphocytes. They have similarities that can cause them to be confused for one another as well. Like we did before, here are some additional examples of each type to show the variation from cell to cell and slide to slide. Both have a single large nucleus, but the shape of it is typically different. The monocyte nucleus is typically irregular, like a ball of clay that has been pushed in multiple places. The lymphocyte nucleus is typically more rounded and smooth. Both have a lighter cytoplasm, but you typically see much more of the cytoplasm in the monocyte than in the lymphocyte. Monocytes also have vacuoles, which can sometimes be seen as lighter circles inside the cells. Another distinguishing characteristic between these two cells is their size. The monocyte is 2 to 3 times as large as red blood cells. This is like the other white blood cells we've talked about so far. However, the lymphocyte is typically smaller than the other white blood cells. The lymphocyte nucleus is usually slightly larger than a red blood cell. Also consider the relative abundances of these two cell types. Lymphocytes are the second most common white blood cell type in humans. They make up 20 to 40% of white blood cells. In contrast, monocytes are much less common, at only 2 to 8%. Finally, let's look at the basophils. They are the least common type of white blood cell, and they are rarely seen in our images. The additional examples provided here are not from our images because we don't yet have many good basophil images from our slides. Even though you're not likely to see any of these cells, you should know their identifying characteristics. Like most of the white blood cells, basophils are 2 to 3 times as large as red blood cells. These cells contain many darkly stained granules around an irregular kidney bean-shaped nucleus. Basophils are very rare, with fewer than 1 found for every 100 white blood cells identified. A version of this summary image can be found in the field guide section of the project. Here we highlight in red some of the main distinguishing characteristics that can help you with their identification. Remember to look for the number and shape of the lobes of the nucleus. Also remember that eosinophils are the only white blood cell with a darker cytoplasm. Lymphocytes are the smallest of the white blood cells. And finally, consider how common a cell type is when trying to decide. We hope this video has shown you some of the ways to determine what type of white blood cell you're looking at. Thank you so much for participating in our project.